Recording begins. It is November 21st, 2025. And this is Biology 105. All right, thank you all for making it. Break's coming. Here we are on the schedule. Week 13, we're going to figure out some, some gene expression stuff. All right, sounds good. We're going to have a vacation. We're going to come back. Genetics, evolution, done. That's it. All right. So you had a lab this week. We did a PCR reaction. There's a PCR quiz. Make sure you complete that assignment. There's a reading assignment. You've been doing this course for a while now. Are there any questions? Oh, the final exam. People are probably concerned about the final exam. That's usual, okay, for that to happen. Study guide's posted. I'm not going to be changing anything that's from what's on the study guide. The final is 33 questions, each worth 3.3 points. All right, and then you get 1 point for free. So it's a 100-point lecture final. And don't forget, in week 15, you have the open lab notebook lab final. Are there any questions about those final things? Okay, cool. Let's get into it. Just let that silence just linger. Usually I keep noise going to increase engagement, but no, not really. I'm really, really excited about it. Yeah. What? Yeah. Oh, yeah. It's good. I mean, more and more I start to just enjoy that. And it's, I don't know, it's nice. But I can only do it for a few days, personally. Yeah. Mm-hmm. Yep. Okay, perfect. Here's where we got to. We're talking about the structure of DNA. We did it. So DNA gets highly condensed. There's a lot of stuff going on here, okay? Something that I haven't really, I've shown you images of it, but DNA, as you go through the cell cycle, it changes in how condensed it is. So during G1 and G2 and the S phase of interphase, right, the chromosomes are pretty much uncondensed. They're just all spread out. Now the DNA is still wrapped around these histones, but they haven't been fully condensed around all of these scaffolding proteins, okay, and other structures like that. That allows the DNA to be read. But if the DNA is wrapped around a histone, it can't usually be read. So that's one way that we can silence or express genes, is just by determining what's actually wrapped around a histone. We'll talk about that a little bit later as we talk about epigenetics, changes to your phenotype that involve regulation of gene expression, but that don't involve any change in actual nucleotide sequence. That's what epigenetics is. It'll come back, okay? So DNA really only gets this condensed down to this form, the standard like 2 sister chromatids that we've seen in the past, okay? That really only happens in prophase. So all these things that are going on during interphase are dealing with this uncompressed, all right, DNA. This chromatin, it's called. But anyways, I just wanted to mention that really quickly. DNA replication, okay? How does DNA get replicated? There were initially 3 main models proposed, all right? One model was that you've got the initial DNA molecule and you read it and you just make an entirely new double-stranded molecule where each new strand is new DNA. So if we look at this image right here, gray indicates the old DNA, all right? Blue indicates newly synthesized DNA. So in the conservative model, those old DNA strands, they stay paired up, all right? You just read them and make 2 new strands, which then pair up. That's not what happens. The other incorrect model was the dispersive model, where you've got the old strand of DNA, it gets chopped up, all right, into a bunch of different pieces, and then the intermediates, or the spaces between those pieces, get filled in. So you end up with this mosaic DNA, okay, where you've got old, then new nucleotides, then old nucleotides, then new nucleotides, and so on and so forth. This figure from the textbook drives me nuts. They've never changed it in any of the editions of Campbell Biology, but these should be alternating. It should be like gray, 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 blue, 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 right? Like, because it's not, anyways, it makes me, every time, it frustrates me. But these just exist so that we can talk about the semi-conservative model, which is the accurate model. So we've got your parent strand of DNA, right? The old DNA, 5' end here, 3' end here, 5' end here, 3' end here, all right? There's the parent strands. You separate them, and each strand is read as a template separately, with a new strand then being built, okay, complementary to it. Because A always binds to T, C always binds to G, you're going to regenerate the same double-stranded structure regardless of which strand you use as the template, all right? And I'll illustrate that here. Oh no, my spiral. I'll illustrate that here with a sheet of paper. How the semi-conservative pathway sort of works. Boom, document camera goes onto the main, please. So one strand of DNA, A, T, A, C, C, G. All right, 5' here, 3' here. Here's the complementary strand, T, A, T, T, G, G, C, 5' end here, 3' end here. If I separate these, all right, and I've got an A, T, A, C, C, G, and then a T, T, G, G, C. Did I do that right? T, A, T, T. It's fine. All right. Each one can be read as a template, okay? It doesn't matter which one you're using as the template because you're still going to generate exactly the same double-stranded molecule. Wait. Yeah, perfect. See, they're the same molecule. Each strand was used as a template. It's semi-conservative. So this is old DNA, this is new DNA, this is new DNA, this is old DNA. And that's the accurate semi-conservative model. Any questions about that? Yes, please. I'm sure it is relevant. I bet you dollars to donuts will maybe we'll touch on it. Maybe it'll come up later. And if it was, you go, oh, you got me. So that's how you can let me know. Okay, I'll do that. Thank you. Cool. So hey, how do they figure this out? Well, they just basically radio labeled more. They just basically labeled the key component. All right. So remember when we were doing the study or talking about the Hervey Chase experiment, where they labeled the protein or they labeled the DNA by using radioactive isotopes. Okay. In this case, DNA's got a lot of nitrogen in it. So you took some bacteria, you cultured them with a heavy isotope of nitrogen, okay? Then you let them replicate, okay? So undergo DNA replication. And then you move them to some bacteria with a lighter isotope. And then you let them replicate again, okay? And the idea here is that you'll be able to centrifuge, okay, the DNA sample and find out if you got all heavy DNA, all light DNA, or intermediate weight DNA. It's a pretty interesting experiment, honestly. Check this out. You let the bacteria divide once, you centrifuge it, there's only one band. What? Well, that's because, let's swap this over to the main document camera view again. I always hit the wrong one. Okay, right here. So the old DNA would be all heavy, all right? The new DNA would be all light. The new DNA is light, the old DNA is heavy, okay? Because the bacteria were grown with the heavy isotope. If you let them divide one time, all the DNA is gonna still weigh the same amount, all right? But following another round of replication, okay, you're going to end up with a heavy strand here next to a light strand. Here you'll have a light strand next to a light strand. Here you'll have a light strand next to a light strand. And here you'll have a light strand next to a heavy strand. Okay? So this is the first division. This is the second division. Right there. Okay? Now, if you look at this image, believe it or not, that's the explanation for these results. And these results are what confirmed that, oh, we've actually got semi-conservative replication occurring, because if you had an entirely new strand being made, you would have had two bands after your first replication, one double-stranded heavy and one double-stranded light. And if the semi-conservative model was what was going on, you would always have one band. It would just over time become lighter and lighter and lighter. Okay? Let me show you the expected results. So the researchers did a good job here. They had expected results, and they knew what those expected results would tell them. All right? Semi-conservative model, that's what you found at the second replication. Predictions of each model. Yeah, what am I doing? I'm just rambling right now. Here were the predictions of the conservative model, the dispersive model, and the semi-conservative model. Okay? The dispersive model and the semi-conservative model agree in their predictions after the first replication, but differ following the second replication. Question? First and second. Yeah.
So when you replicate DNA, then you've got some DNA that's older and some DNA that's younger.
Yeah.
Does that mean, like, at the end of meiosis, you've got potentially like a haploid cell that has older DNA than other like sister cells? Does that make sense?
Yeah, no, I understand what you're saying. So because the cells before they undergo meiosis are undergoing rounds of mitosis, right? Yes, some of them could have certain certain chromosomes that have more old DNA than others have more old DNA. But the age of the DNA, it's not necessarily like those individual nucleotides, and their age isn't necessarily very important to their function. It's more important for figuring out how the replication occurred, if that makes sense. The old DNA strand's not necessarily worse.
Okay.
Yeah, awesome. Yes, please.
So basically, correct me if I'm wrong, but this is based off of density?
Mm-hmm.
Okay. So the old DNA.
Well, yeah, because it's like mass per unit volume.
Is heavy.
Yeah.
The newer DNA is lighter, so the lighter DNA would be at the top, and the older DNA would be at the bottom.
Yeah, and these were centrifuge samples, so
Okay.
heavy DNA goes down.
Yeah, so they figured out that because of density,
Essentially, yeah. Yeah, awesome. I love it. Cool. So DNA replication. When does DNA replication occur? In the S phase of interphase. That's when there's new DNA synthesis. Okay? Solid? In E. coli or in prokaryotes, the DNA is simpler. There's less of it, all right? Prokaryotes have a single chromosome. It's a circular chromosome, so it's joined end to end. And DNA replication is therefore pretty different than in a eukaryote, where you've got a ton of different linear molecules, all right, making up each chromosome, and there's way more genetic material. In prokaryotes, okay, it's pretty straightforward. The DNA separates at a certain spot called the origin of replication. The parental strand, the old strand, is read as a template, and the new strand is assembled. The complementary bases are just attached. This keeps expanding out. These strands, these parental strands, keep getting separated with the new strands being added, okay, base pair complementarity. Keeps expanding, and eventually you get two daughter DNA molecules that are half old DNA and half new DNA. Here's an actual image of this occurring. You can sort of imagine that this is going to keep stretching out, and eventually they'll just snap off from one another. So because the chromosome is so small, and because it's circular, the process looks a lot different inside of prokaryotes. Now, what's actually going on at the replication fork, that's very similar between prokaryotes and eukaryotes. What really differs more is there's a lot more DNA in eukaryotes, and you can't just replicate it all with one origin of replication. Okay? So in a eukaryotic genome, you've got a bunch of origins of replication, okay, where these replication bubbles can form. The strands get separated and complementary bases get added, reading the parental strand of DNA. You just keep adding complementary bases. Then all of these replication bubbles sort of just grow together, and they're all joined, and now you've got two daughter DNA molecules, each one of which is half the original template DNA and half newly assembled DNA. Cool? All right, awesome. So the big question is, what's actually going on at the replication fork? So the process that's going on is, as you know, is I'm going to read that old strand, and I'm going to add the complementary bases. If I see an A, I'm adding a T. If I see a T, I'm adding an A, and so on and so forth. That's what's going on, right? And what's going to be responsible for this? A bunch of different enzymes, okay? It's always enzymes, all right? Enzymes catalyze chemical reactions, and what's happening here is an anabolic reaction, okay? Where a new molecule is being assembled, you're just reading something as a template to direct the sequence of nucleotides. So we've got the key enzymes, the helicases, the single-strand binding proteins, the topoisomerase enzyme, the primase enzyme. Sorry for the slight formatting issue that threw primase down on the bottom there. You can see that this slide, it's a little worse for wear after my last reformatting. Let me just, make it worse. There. No problem. Okay. So, helicase, it's like the zipper, or the key, I believe is what it's called. Anybody here a zipper expert? The name for the thing that separates and pushes the two ends of the two parts of the zipper together. I'm pretty sure it's called a key, all right? It connects the teeth. Either way, there's a name for that, and that's what the helicase enzyme basically is. I wish I was wearing a jacket with a zipper, because I would go zip, 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 helicase, all right? But just theater of the mind. Just imagine. Zip, zip, look at the helicase. Zip. All right. And I'll come back to reality. All right, back here with me. I know. Whoa, where were we? All right. So once you've separated those parent strands of DNA, they're perfectly complementary to one another, right? DNA is pretty stable. So just pulling these things apart, it's like having two strands of magnets. They want to snap together. So if I just pull them apart and then let go, they're just going to snap back together. So there are these single-strand binding proteins, and what they do is they bind to the separated single strands and just prevent them from snapping back together. All right? Because this is an A and this is a T, and they want to come together via hydrogen bonds. Okay, great. Topoisomerase. Now, topoisomerase is a tiny bit confusing to think about. This is another one where I used to illustrate it with my headphones, but this will work right here, okay? It's not really on the screen, so those of you watching the recording, you're just gonna have to imagine this. Check this out, right? I've got this double helical structure of this twisted cable, and then I start to separate the parent strands. What's happening? It's overwinding up here, all right? It's building up all these problems. The DNA is gonna get all jumbled. It's all messed up, right? So what topoisomerase does is effectively, it just makes a little cut here, and then it lets the DNA go whoop, and then it makes a little cut and goes whoop to relieve that tension so that the DNA can continue to open, all right? Sorry for those of you watching the recording, but everybody here can agree it was an incredible demonstration. Topoisomerase, thank you. It relieves overwinding, okay? Now, primase adds an RNA primer. We're going to talk a lot about why the RNA primer is essential, okay? But this RNA primer needs to be there for DNA synthesis to happen. You need the RNA primer present so you can add new nucleotides to that end, okay? We're going to talk about it a lot, so I'm just going to ask that for now, just know that primase adds an RNA primer, and you need that to add new nucleotides. Okay, cool. Anti-parallel nature. I have told you multiple times when we're talking about the anti-parallel nature, we are really only worried about the 5' and the 3' ends, okay? Not about base pair complementarity, and we're not saying that this strand is the same as this strand running in the opposite direction, which is usually the misconception about anti-parallel nature. Instead, it's all about which end has the 5' and which end has the 3', and that's because of how you energetically add new nucleotides to a growing polynucleotide chain. You're doing, right here, you're building something larger from small subunits. You know that's an anabolic reaction. You know that's going to be endergonic because you're going from disorder to order. You need to have some energy available to allow this to happen. So how it happens is through the use of these nucleoside triphosphates. ATP is basically just this, all right, when it's free. This is like NTP, all right? Or this would be TTP, all right? This would be like GTP and CTP. What does that mean? That means that the free nucleotides have been energetically charged. But that energy is available on the 5' end, all right? So breaking these nucleotides off, breaking these phosphate groups off is going to give you the energy that the enzyme needs in order to assemble the growing strand. But because these phosphates are on the 5' of the new nucleotide that's going to be added, you can only add them to the 3' end of the growing nucleotide chain. So energetically, you have to synthesize a growing strand in the 5' to 3' direction, adding new nucleotides to the 3' end. Because if you add a new nucleotide to the 5' end, the enzymes can't do it. There's no energy available for that process. Question? You add to the 3' end, yep. And so what we say is that DNA synthesis occurs in the 5' to 3' direction. New nucleotides are always added to the 3' end. Yes, please.
You definitely said this at some point, but could you remind us why it's called the 5'?
No, I won't do that. I refuse. Yeah. Thanks. Yeah, it's super mellow. Check it out. Here's a ribose sugar. And if you were numbering the carbons here, you would number this as carbon 1, 2, 3, 4, 5. All right, so the 3' end is right here, where the hydroxyl group is attached to the 3' carbon. It's all carbon numbering. And when I first started to do this class, I used to do carbon numbering talks that this would make more sense, but I found that it didn't pay enough dividends. It's like, why are we worried about carbon numbering's annoying, you know? Yeah, please. The 5' end of this is being attached to the 3' end here. Yes, exactly. Yeah, you add them to the 3' end because the energy is available right here. And it's totally fair to be like, well, wait, why couldn't we do it the other way if you were adding to the 5' end? Why couldn't it just have the phosphate groups on it and you break that off and build it in the other direction? Because the bond here has a bunch of energy. When you break that bond, you release a bunch of energy. Yeah. Of course, no. Yeah, absolutely. So usually we just say, hey, let's just memorize this 5' to 3', but I want you to understand a little bit about, like, you're doing an anabolic reaction here. The energy has to come from somewhere. The energy is in this nucleoside triphosphate, and you're coupling an exergonic reaction of breaking those phosphate groups off with an endergonic reaction of forming a new covalent bond there. So it comes back all to our how do enzymes work and how do energetics work, and everything has to follow all of the rules, or else the rules are flawed. You know what I mean? I'm not making good predictions. Anyway, let's get into the most challenging concept in genetics for most students, and that's the idea of the leading and the lagging strands. I have tried so many ways of explaining it, but what's really worked is me preempting it by saying, this is what everybody struggles with. So it's totally normal if you struggle with this concept, and we're just going to go through it, ask lots of questions. Okay? Here we go. You need the RNA primer, okay, because DNA synthesis can only occur in the 5' to 3' end. So what the RNA primer does is it effectively provides a 3' end. Let me double back a little bit to right here. See this RNA primer? You have to have one, okay? So you have an exposed 3' end so you can attach new nucleotides. Once you've got a couple of nucleotides present, then you can just keep the chain reaction going, right? Here's the problem. This strand, the 3' end, is in the same direction as the separation, okay? So you're going to keep having new template DNA exposed on the top here, and you're going to be able to continuously make new, make a complementary strand, okay, as a separation occurs. But over here on this strand, if I've got a primer right here, all right, then synthesis is going to go in this direction, which means that eventually new template DNA is going to be exposed, and I'm going to have to attach a new primer. And then I'm going to have to attach a new primer. And then I'm going to have to attach a new primer and keep running into the previous primer, okay? Additionally, these primers are made of RNA. And if my new strand of DNA is going to be identical to the initial strand of DNA, I can't have RNA nucleotides in there. It has to be all DNA nucleotides. Because remember, RNA and DNA are different. There's a different sugar involved, for example. Yes?
I was about to say, isn't the difference between RNA and DNA the U enzyme, the U nucleotide instead of the T nucleotide?
That's a huge important difference about it too. Yeah, there's uracil instead of thymine. So if there should be a T, a DNA T, now there's a totally different nitrogenous base in there, which has different behaviors as it interacts with the adenine nucleotide. It's less stable, for example. So if you left the RNA primer, you would be making this mosaic daughter strand that has RNA in it and probably has lower stability if there's any uracil present, all right? But at the very fundamental level, this process makes copies. And if I'm separating these and I'm reading and I'm not pulling the RNA out, then I'm not making true copies. I'm taking a template and I'm making half old template and half new with RNA chunks in it. Now for a limited time only with RNA chunks, all right? It's a very limited time because there's an enzyme involved in removing those RNA chunks. Just like when I get an ice cream that has toffee bars in it and you excise those with, a spoon, then you leave the ice cream. The analogy falls apart pretty quickly, but that's fine. That's fine. Check it out. This is what I was just talking about. Here's an RNA primer with the 3' end on the left side. So the new strand is being synthesized in this direction, and as new template gets exposed, you can just keep growing continuously. This is the leading strand. It's synthesized more rapidly because it's a continuous chain reaction process. So it just keeps going until you reach the next replication bubble, of course, where then there's another primer that you run into, but blah, 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 In these lagging strands, what's going on is, check this out. This is the primer with a 3' end on the left side. Let me just, I forget sometimes that I can zoom in like so. Okay? So this is a primer with a 3' end on the left side. It's been put as close to the replication fork as possible, and the new strand is built in this direction. As new template DNA is exposed, you got the leading strand on the opposite end just going and doing its thing, but you have to add a new primer here and then run back to this primer. So that's the lagging strand. It's slower because you have to keep waiting for new primers to attach and for new template DNA to be exposed. And then you have to cut out all of these RNA nucleotides, replace them with DNA nucleotides, and then join the strands together. So that's the lagging strand. It lags behind. It takes more time. Okay? Let's look at another image, which my solution last semester to how complicated this is was to have like 4 different slides that all showed the process in slightly different ways. We'll see if it works this time. Please.
The light strand RNA goes towards the 5' end, and it goes backwards, to the right.
So I'm just gonna try to like re-explain it. Check this out. The template strand, right? There's a 3' end here, 5' end opposite, 5' end here, 3' end opposite, right? So when I attach an RNA primer to this strand, the 3' end is going in the same direction as the replication fork. I think this is what you said. I'm trying to okay, cool, awesome. In the other strand, the 5' end is over here, right? So whenever I add a new primer, the 3' end is going to be facing in that direction towards like the end of the DNA molecule. Does that make sense? The new DNA replication has to always start at the 3' end and go in that direction. But it's going to be on the opposite of the 5' end of the template strand, where it ends up. Does that make sense? Awesome. Yes, please.
So for the original 3 and 5' end being like this direction, that's when we always appear lagging, or does it change it? And then the 5' is always here leading.
Oh yeah, I totally see what you mean. So if the replication fork is moving towards the 3' end of that template strand, then that's always going to be the lagging one. If it's moving towards the 5' end of the template strand, that's always going to be the leading one. Yeah, that's what you said. Awesome. Perfect. Yes is the answer to the question. Great. There are a lot of different ways of thinking about this, folks. And, whatever model works for you to help you understand it. But check this out. Here's the other problem. You got to remove these RNA primers. And once they get removed, and even once they're replaced with DNA nucleotides, all right, you then have to join those chunks together, because that can't be done by the regular enzyme that just builds the new nucleotides by adding new nucleotides to the 3' end of a growing chain. So now there's an additional enzyme that needs to be involved here. It generates these chunks of nucleotides, okay, that aren't joined to anything on either end, and they're called Okazaki fragments, okay, named for the researcher who discovered them. So those Okazaki fragments need to be joined together. Here are the Okazaki fragments in the smaller diagram. Here are the Okazaki fragments zoomed in, okay? Good. Question?
So you have the RNA primer, and it's produced that you have multiple of them producing DNA as, DNA replication process takes place. When does the new DNA, the lagging strand, how does it know, or how does it stop when it hits a previously made DNA strand, because it's made in chunks?
Yeah, absolutely. So once the DNA polymerase, which is the enzyme that makes a new chain of DNA, it polymerizes DNA. It's DNA polymerase. Also, there are a few different types. That's why I haven't had a slide about it yet. But once the DNA polymerase identifies that once it's no longer a single strand, it cannot attach to it. So the DNA polymerase just falls off. It won't bind to a double-stranded DNA molecule. So that's how it like knows to fall off and stop synthesizing new nuclei. Or not new nuclei. Stop synthesizing a new strand. Fair enough? All right. And then we got the DNA ligase responsible for joining those fragments together. Okay. In the leading strand, you get one RNA primer, and then the DNA polymerase attaches nucleotides in the 5' to 3' direction. In the lagging strand, the DNA synthesis is moving away from the fork, like you'd said earlier, okay? And so you have to keep adding new RNA primers. Cool. The RNA primers get removed by a different DNA polymerase, all right? And there's an enzyme called DNA ligase. I'll talk to you about the different DNA polymerases, don't worry, but they all just have names like DNA polymerase 1 and 2 and 3 and 4, okay? And of course, they're not named in the order that they operate. They're all named in the order of their discovery, so the numbering system's not very useful for understanding the process. Okay. Good stuff. Zoomed in again, different image. I'm going to try not to be too repetitive, so I'm just going to keep moving forward unless folks need help. Remember, like I said, people struggle a lot with the leading and the lagging strand concept. So if you're still not getting it, that's not a problem. That's why I'm here. That's why I make the big bucks. That's why I get to drive a 2000, 16 Forester? You know? I don't want to brag, but yeah, parents gave it to me. Yeah. Pretty cool. When my RAV4 had a head gasket blow. That's a whole story. You want to talk about that instead of DNA replication? Yes. Yeah, me too. Yeah, please.
Just to clarify, if the replication fork is moving towards the 5' end is the leading one.
If it is, so.
Like if the replication fork is breaking towards the 5' end, the leading one is going towards the 5'.
It's better to think about if the newly synthesized strand is moving towards the replication fork, rather than thinking about the end of the template DNA. In the leading strand. In the lagging strand, it's moving away.
And when it moves away, it moves in fragments.
Yep.
You have to have new RNA primers put on it each time, and then those RNA primers need to be taken off by the polymerase, and then the fragments need to be joined together by the ligase.
Exactly. Great summary of the whole thing. Yep, nailed it. No notes. Of course. Was there another hand up? I see your hand. Okay, cool. This is great, folks. Yeah, this just shows you the process again that we've been talking about. Good stuff. Good stuff. Single-strand binding proteins, DNA helicase, topoisomerase. We already talked about them. There's just another image of that. There's an embedded video that you can watch if you want to watch DNA replication videos. I'm so sorry, everybody. It's great to know that that's how I react when I'm panicked, you know? Good, yeah. And it's immortalized in a recording. Okay. Oh, whatever. We'll just leave it in there. We'll keep it in there for posterity, right? Let's go ahead and talk about the different enzymes now in table form. I don't know. I always like a good table that just organizes everything together into one place. So let's just run through these functions real quick. You let me know if you got some questions. Helicase, it's the zipper. It's the key part of the zipper. I'm just going to assume that it's called a key. Single-strand binding proteins stop the template strands from snapping back together. Topoisomerase relieves overwinding strain. Primase adds the primer. Okay? Now, we got the two different key DNA polymerases here. There are more DNA polymerases, of course, right? That are, bless you, that are involved, say, in checking your DNA. Like these DNA polymerases, some of them are involved in the checkpoints of DNA, of cell division, okay, where they scan your DNA and they make sure that there aren't any mismatches. DNA polymerase 1 is the one that removes the primers, all right? DNA polymerase 3 is the core DNA polymerase that's synthesizing the new strand, reading the old strand as a template. All right. So DNA polymerase 3 is the one that's synthesizing the new one. It's reading the old strand, saying there's an A, let me put a T here, let me put a C here, let me put a G here, always going in the 5' to 3' direction. DNA polymerase 1, okay, it's kind of weird because what it actually does is it snaps out these, it snaps out the RNA, it snaps out the primer, okay, and then it adds the new nucleotides to the 3' end of the adjacent fragment. So it pulls these out, and then it has to still operate like a DNA polymerase. So it adds the new ones to this end here. That's not important for our purposes, but if you want to see that everything's still following the rules. And then ligase joins the new strands together, okay? And those are the key players in DNA replication. Are there other things happening? Of course there are. But this is a pretty good, baseline understanding. And hey, if you're already reading your dang Stuff of Life graphic novel that I've been recommending so much, you get to see all of these players as fun cartoon characters. Well, ends of DNA molecules, all right? Telomeres. Heard about telomeres, maybe? I'm looking around the room. I'm making eye contact with a number of students, and I'm seeing nodding and recognition. I'm seeing shaking of the head. We will talk about telomeres. Basically, every time a DNA molecule, a double-stranded DNA molecule gets replicated, you end up with this single-stranded chunk of it. Because check this out. Look at here's the strand. It finally reaches the end of the chromosome, okay? Now, on this template strand at the bottom, where the 3' end is exposed, sure enough, you just attach an RNA primer, snap it on right there, okay? And that's fine, right? You can go ahead and make everything, make all the template, join all these Okazaki fragments together, you're good to go. But here's the problem. When you remove that last RNA primer, if you remember how DNA polymerase 1 works, there's no 3' end to add new DNA nucleotides to here, because you're at the end. So you excise that RNA primer, and you end up with the end of the DNA strand being, single-stranded. There's a problem. It's no good, all right? Now, this may be very important in processes of, I was just reading something, it might be very important in processes of homologous chromosome pairing, actually, which is pretty fascinating. It's a whole rabbit hole. But for our purposes, what it means is that DNA is constantly shortening. Because look, now when you take this strand and you separate it again, this template strand is now going to have a complementary strand attached to it, and it's going to be shorter. And this strand, you got another, single strand portion that didn't get replicated. So DNA gets shorter and shorter and shorter. Chromosomes get shorter and shorter and shorter every time they replicate. Okay? So how do eukaryotes deal with this issue? Prokaryotes don't have to worry about it because of their single circular chromosome. But eukaryotes, we've got chromosomes that are constantly shortening. Okay? Once the chromosome reaches a certain length, triggers apoptosis, okay? The cell starts to cut into essential nucleotides that are encoding proteins. So there's this region at the end of your chromosomes called the telomere. And the telomere has just a bunch of nonsense, non-encoding nucleotides. They just exist so they can keep getting shorter and shorter and shorter, and it's not going to have any impact on the actual important instructions in your DNA, which are all like over here, all right? So the telomere just keeps going shorter, shorter, shorter every round of division. So this is one mechanism by which aging occurs, because your cells can only divide so many times before eventually the telomere reaches its end. It's like a fuse, burning down. And then the cell either undergoes apoptosis or it just enters into a non-dividing stage. So now the cell's there, it's still doing its job, but it's just getting crummier. It's getting all gunked up. Everything's kind of falling apart in it, This is one mechanism by which aging may occur. So why don't we just have an enzyme for lengthening the telomeres? Because there are enzymes that exist that lengthen telomeres. Telomerase is the enzyme, and it's active in germ cells, because when you've got like your germ cells dividing, you don't want to be making sperm cells that already have short telomeres. You don't want to be making egg cells that already have short telomeres. So there's an enzyme active to lengthen those telomeres. Why don't we just express it in the somatic tissue? And one of the main reasons is when this does happen, you end up with a lot of cancer, okay? There's lots of questions about aging. Is aging actually beneficial for a population? Because natural selection acts on populations, not on individuals. Dying of old age is detrimental to the individual. I don't want to do it at all, personally. So you let me know when you get your telomerase pill working, and I'll take that pill, okay? I'm going to live forever via my students who will someday invent longevity-increasing pills that I can take. So get to work. We'll talk about this more on Monday. Have a wonderful break. Thank you all so much. And give me that pill. Make me that pill, all right?